I would say we have a lot of the standard optical imaging cores that you might see at other institutions, and that might be a fluorescent microscope and things that we call a confocal or two-photon microscopy. I think part of what we also offer is customized microscopes that will be specialized for like in vivo imaging, so we can actually look at physiology and behavior of cells, single cells, in the whole animal. We can also look at single molecules, so we can go from the point where we're looking at very minute signals inside single cells all the way up to whole animal imaging. We have a broad service. I think that's one of the advantages of the imaging core. It's not just one instrument, it's actually multiple instruments that we take advantage of.
The highest resolution system that we have is called super resolution, so you can go down to 20 nanometers. That's 10 times better than the conventional optical microscopy, which will give you a resolution around 200 nanometers. But resolution is not everything, so sometimes if we want to do, say, image deep into a tissue to know things happening deep there, so then we will have to give up some resolution for that kind of study.
We've got several applications. One that I think is quite interesting, it sort of involves many of the different microscopes or techniques that we use. It's looking at the growth of single cancerous cells in the brain, so it's actually gliomas, a brain tumor. It's work from Dr. Brenner's group. And basically what they do is insert a single cell, a cancerous cell inside the brain of a mouse, and then you can watch it grow over many weeks and months. And during that time, what they're really interested in is the vascularization that actually will grow into the tumor, so to help support the tumor grow. And they've been studying different reagents then that will slow the actual vascularization, so to control the cell growth or the tumor growth. So in that particular case, it's more on what I'd call a whole organ level, if you want to call a tumor sort of a whole growth level. On other levels, we might be interested, say, in a single cell, and we're interested sort of in the physiology. So a particular drug does something, like we're interested in understanding how an agonist might be a better neuroprotectant, say, after stroke. And so we think the way that drug works is to increase calcium. That's actually a calcium you know a lot in your bones, but actually it's something that's released in a really important signaling molecule inside cells, so we can actually have probes that will light up more brightly when calcium is bound to it, so we can get in real time then changes in intracellular calcium as sort of a fluorescent signal, and so we can record that again over a temporal sequence of time, maybe over minutes. This is kind of projecting into the future a little bit. We're now starting to utilize what are called Grin lenses, a particular type of cylindrical piece of glass that acts like a lens, and we can actually then insert that deep into the brain, so it's more like a probe. Think of it as a light microscope that we can sort of insert fixed into the brain, so we can look down into the brain centers. These are things that we're just learning how to develop with actually some collaboration with other laboratories. So we're hoping to sort of extend some of the work that Dr. Wang's helped us out with, deep into the brain too.
And so another part that I can share my expertise is that I help people to quantify their data. So a lot of what we'll do, the services that we offer, we actually have to sort of fine-tune it, tailor it to the individual's needs, and so we'll sit down and consult with them and see what do you want to do, and then we'll decide what you can do, and then we'll try and see if it can be done.